So today we will discuss about translation of eukaryotes. Earlier I taught you about translation of prokaryotes. Is there anybody who has read it? Is there anyone who has read translation of prokaryotes? Then it will be very difficult. What I thought that basically I will skip all those things whatever I have told you in prokaryote translation, because the basic process is almost same, whether it is in transcription and translation of prokaryotes or translation of eukaryotes. Only some important points are there, differentiating points that you have to remember and you have to memorize. There is no way out. Of course, the translation of prokaryotes was much simpler, and usually translation on prokaryotes are coupled to transcription because there is no separate compartment between nucleus and cytoplasm. But in eukaryotes, transcription occurs basically in the nucleus completely, and then it is transported to the cytosol. That's why translation in eukaryotes are totally different. It doesn't occur simultaneously, just like prokaryotes. It occurs in the cytoplasm mainly, and there is a separate structure of translation machinery for translation within the mitochondria, that is called mitochondrial translation. After the protein has been made, all the proteins are not functional in that stage. They need to be modified with the addition of something or with the cleavage of peptide bonds or certain portion of the peptide. These are all called post-translational modifications, and you have to know all these things because any of the structure, any of the things might come in the question as a short answer question, long answer question, even at times MCQ also. So basically one question may be asked on process itself, another question may be on post-translational modification. One thing I would like to know, since we have been told about ribosomes, and in the initial part during cellular organelle and cellular suborganelles, ribosomes were told. Can anybody tell where ribosomes are formed basically? Is there anyone who can tell where ribosomes are formed? Where it's a cellular organelle, basically subcellular organelle I would say, because it is present within the cytoplasm. But where ribosomes are synthesized? The ribosomes are made of ribonucleic acid and protein. Where they are synthesized? Any idea? Any idea? See, ribosomes are synthesized within the nucleolus, and they are also assembled at the nucleolus or in the nucleoplasm, or they can come out through the nuclear pores into the cytoplasm. So formation takes place within the nucleus, assembly can also take place within the nucleus and nucleoplasm, and mostly it is also in the cytoplasm itself. So the prokaryotes we have said, you have learned basically that this is 70S ribosome. Why S is called, that I have explained. I am not going to do that thing. Now eukaryotic ribosome is of 2 units again, but it is of 80S. It is of 80S. This 80S is having two units. One is smaller unit, that is 40S, and the larger unit is 60S. Now this is having three ribosomes. One is 20S rRNA, 5.8S rRNA, and 5S rRNA, plus about 50 proteins, approximately 50 proteins. So this is the part of this one, and this 40S basically, remember, try to remember, it is 28S and it is 18. Only one rRNA and about 34 proteins. 34 proteins. So basically, these ribosomes are having a scaffold of ribonucleic acid, rRNA, on which proteins are placed, they are interacted. So they form a ribonucleoprotein, and this is the component. Normally, these both the units are bound to each other. But when they are bound to each other, in that stage, protein synthesis will not occur at all. They need to be dissociated into two units, and then initiation will start after the formation of complete 80S ribounits. This will be larger fragment and the smaller fragment. Basically, the smaller fragment, that 40S ribosome, maintains complementarity with the mRNA molecule. See, ribosomes are present as free form in the cytosol and also present as bound form with the smooth endoplasmic reticulum, that is called rough endoplasmic reticulum. So protein synthesis occurs in both ribosomes, free ribosomes and bound ribosomes. Free ribosomes synthesize those proteins which do not have basically a signal peptide, and they are required for intracellular functioning, like enzymes, like intracellular structural proteins, but they are not usually exported or transported elsewhere through a secretory vesicle. So those proteins which are having a signal peptide and they are required to be excreted or transported through a process of exocytosis are produced within the rough endoplasmic reticulum. So you must remember that thing. So ribosomes basically are produced within the nucleolus and then assembled either in the nucleolus, nucleoplasm or cytoplasm. Ribosomal proteins enter the ribosomes at a speed of about 1,40,000 proteins per minute, and from the nuclear pore of about 200 nuclear pores, 2,000 ribonucleoproteins are passed through. That means 70 times less than what is entering. So 1,40,000 proteins is not a matter of joke. So this much protein has to be produced within the cytoplasm, and then they are transported to the nucleus. And in a growing cell, the number of ribosomes are about 5 to 10 millions. So, considering that 10 million ribosomes, about 70% of the ribosomes are present within the as free form, that is almost 7 million. So that 7 million forms the protein of those which are required for intracellular functioning. Rest 30% are for others. So ribosomes can also be defective because of ribosomal RNA or ribosomal proteins. So they can produce disease that is called ribosomopathy. These are very uncommon, but one must be aware of, particularly those who are trying to opt for pediatrics, because they will present this type of diseases, genetic diseases at a very early stage, and they might camouflage the symptoms of other diseases. So ribosomopathy, try to remember that one disease is called ribosomopathy. Second, translational machinery. I told you earlier also that this requires ribosomes, tRNA, rRNA, mRNAs, and several other proteins known as factors called initiation factor, elongation factor, and termination factor or release factor, basically they are said. And what are the initiating factors, elongation factor and termination factor for prokaryotes, eukaryotes and mitochondria? That you have to memorize, and what are their functions. Second, all ribosomes are having a conserved catalytic center, means there are two terms, one is conserved, one is consensus. Conserved means that sequence of nucleotides which has been preserved or conserved during evolution. So it represents an evolutionary origin. So that conserved sequence basically is required for the survival. So if by chance there is a mutation, that will cause basically a major problem. And here in the translational machinery, the catalytic center, that is peptidyl transferase center, is present within the larger subunits, whether it is a prokaryote or whether it is an eukaryote. That it acts as a catalytic site, and that is responsible for peptide bond formation. Now, always ribosome during mRNA synthesis, during protein synthesis, mRNA accepts the activated tRNA. Activated tRNA means where it has been activated this amino acid with the tRNA through a process of hydrolysis of AMP, ATP got converted into AMP, and two equivalent amount of high energy compounds are released. So both ATS and 40S are having dozens of proteins. These two units are joined together through insertions of protein projections. So these 40S, and during prokaryotic translation, I told you that 40S ribosome is having, suppose in this case, is 18S rRNA is there. So mRNA will interact with this rRNA. This is rRNA and this is mRNA, which will come. So they will maintain a double stranded structure of other than molecule itself. So they will position such that the initiator codon is placed at the P site. Once they are formed, this ribosome site, this larger and smaller subunits, they are having three sites. One is called A site, that is acceptor site. One is peptidyl site, P site, and third one is exit site, E site. That is the site for the free tRNA to be released, and this is the site for entry of the incoming amino acid tRNA. Remember, out of these three sites, This is E, this is P, this is A. During initiation, the first amino acid is positioned at P position only, first amino acid. That is initiator amino acid. This is positioned only at the P site. Rest all during elongation phase are positioned at A site. That is the importance of P site. And this P site is basically attached, the peptidyl tRNA is attached here. Growing chain is incoming amino acid tRNA, all subsequent during elongation process will occupy this site, and this site is blocked during initiation stage by initiation factor 1A. This site is blocked so that the incoming this first amino acid cannot be placed at the A site. Now, once this protein synthesis is complete, then this ptRNA is positioned here at the exit site from where it goes out. So basically, I repeat that one thing, that first positioning this is a very important step. Initiation of doing anything is really difficult. So there is pre-initiation complex and initiation complex that has to be formed first, and then only initiation will occur. And the main purpose of initiation complex is to place the first codon, starting codon right at the P site properly, and the reading frame of the mRNA is maintained with all initiating factors. And once the unit is totally formed, this complete unit 40S and 60S, then the factors are released and the elongation factor comes into play, and elongation takes place. And during elongation again, there are three steps. One is incoming amino acid tRNA, then peptide bond formation, then release of the tRNA, translocation of ribosome 1 codon to the 3' side, and this process continues and the cycle goes on, and at the end of protein synthesis, both the units, subunits gets released to form again a new RNA formation. So first and foremost thing that these ribosomes need to be dissociated. Basically, the ribosomes are kept either in association stage or in the dissociation stage by the initiation factor. Initiation dash I will discuss also. So individual mRNA molecules are having a different rate of translation because of the regulatory elements present in it. What are? So this is in short. I'll discuss about the cycle in detail. See, these salient points you note down. One is initiation. Translation occurs mainly in the cytoplasm. Mitochondrial translation occurs for mitochondrial needs mainly 13 numbers of enzymes which are required for the electron transport chain. All other mitochondrial proteins are synthesized by the cytoplasm. That is under the direction of nucleus. So initiation is basically ribosomal dissociation is a mandatory step. So as I told you, after the protein has been formed, ribosomes get dissociated, and in order to use those dissociated ribosomes, they are kept in the dissociated state, and they can start another cycle of translation. So first ribosomal dissociation, then ternary complex formation. What is ternary complex? I told you earlier that activated tRNA, elongation initiation factor 2 number 1 here, and the GTP. So basically GTP initiation factor 2 binds to GTP. So that is a binary complex that combines with the activated, that is amino acylated tRNA to form a ternary complex, and then this ternary complex binds to the 40S ribosome along with other factors to make 43 pre-initiation complex. And when this 43 pre-initiation complex combined with the mRNA, then this is called 48 initiation complex, and that is ready for. So elongation is basically, so remember ribosomal dissociation in the first stage, second stage is ternary complex formation, and third one is mRNA activation. What is mRNA activation? Means again addition of factors 4F and 4B, and there is a protein called poly-A binding protein. You must remember that the structure of the mRNA was having, modified post-transcriptionally with the addition of poly-A tail. So the poly-A binding protein will also be there, and 4F and 4B will be added. And there is also a 5-methyl guanosine cap. So some translations are cap dependent, some are independent. Normally 7-methyl, this 5' capping, cap dependent translations are is made only in special situation when the cell is under stress. At that time translation process is inhibited, and then 5' cap based translation doesn't occur. Instead, that cap independent translation occurs, and in that case the initiation takes place directly through the internal ribosomal entry site, or it can occur through the direct binding. So elongation will, as I told you, that first codon, first amino acid is placed at the P site, and then the attachment, subsequent attachment is made at the A site, peptide bond formation is there, and ribosome translocates by one amino acid. It's basically an energy intensive process. So formation of one amino acid tRNA will cause wastage of 2 high energy phosphate bonds. Now placing one activated amino acid tRNA to the A site will again cause one molecule of high energy compound, that is GTP to GDP, and from displacing after peptide bond formation from A site to P site, again one more GTP will be required. So to make a peptide bond formation, basically 4 high energy phosphate bonds are required. So it's an energy intensive procedure that you must remember. So termination, once the mRNA reaches, mRNA is having a start codon, and that is recognized because there is no tRNA for that. So that is, so no more elongation takes place. So translation stops at that point, and then release factor comes into the play. That is, in this case, it is a universal release factor RF1. RF3 also releases RF1, So RF1 and RF3 are the main this elongation termination factor, and if time permits, I will discuss about mitochondrial translation. What is that? So try to remember the difference between the two. I mean, prokaryotes and eukaryotes. See, prokaryotes are having 7 Ts, eukaryotes is having 8 Ts. Subunits 50S, 30S, subunits 60S and 40S. Since today we are considering this, we are discussing this eukaryotes, so it is having a 50 proteins almost, and you can see there are 3 rRNAs, 28S, 18S and 5S, and here it is 18S, and there 34 proteins. So the amount of nucleotides, you can see the amount of nucleotides is 28S, that means it's a bigger size. So it is having a 3354 nucleotides. The smaller the size, here the 5.8S and 5S, you see the difference is 0.8 only, and there are 2 nucleotide difference, 154 and 120 only. Chalo. So there are several factors which are required. So IAE is prefixed initiation factor. For the prokaryotic factors, we do not have any prefix, but for eukaryotic, we are having a prefix EIF or EEF, means it is related to eukaryotes only, not prokaryotes. So our ribosomes are kept dissociated by these 2 factors. This is dissociation, it is EIF3 and EIF6. If they are bound to that subunit, small subunits, then there will be no association. So they will keep the ribosome in the dissociated state. Similarly, reassociation takes place with the release of those factors and binding of E15, and that causes reassociation. And you can see the initiation factor here. There are at least 10 to 12 initiation factors are required to start this initiation complex, and the elongation factors are basically 2, elongation factor 1 and elongation factor 2, and release factor, universal release factor is release factor 1. So there are different functions of these initiation factors. All these you have to remember from the book. There is no way out. One important point I must say that this initiation factor 1 and 1F binds to the 40S unit first. That is number 1 point. Number 2 is E1F3, EIF3 basically. Initiation factor 3 is the largest initiation factor, and the control of initiation is made by 2 factors, EIF2 and EIF4E. EIF4A is having a dead box. What is dead box? See, 1-letter abbreviation of amino acid, if you recall, then D represents basically aspartic acid. D represents glutamic acid. D represents aspartic acid, E represents glutamic acid, A represents alanine, and again D represents aspartic acid. So that particular initiation factor is having a dead box, that sequence is there, and mRNA is having a helical structure. So that helical structure needs to be removed. So that means that there must be winding of that mRNA structure, and that is done by the EF41 and EF4E, that recognizes the 5' capping of mRNA complex. Next. So there are homologs and analogs. Homologs are the structures which are of common embryonic origin. But analog is basically not of embryonic origin, but they are functionally similar. So that is the difference between the homolog and analog. So we are having prokaryotic analog initiation factor 1. It is corresponding to initiation factor 1A, because that is occupying the A side during initiation, not to allow the any incoming amino acid to bind to this site. IF3 is EF1, that is acting almost just like that first factor, and the EFP is equivalent to EIF5A, and analogous structure is like this. Now dissociation, as I told you, there are two dissociation factors, one is IF3 and IF6. Association factor is one, that is EIF5. So if the amount of the factors are present in excess or less amount, the cell will not be able to function properly. As you can see that in cancer, EIF3 is overexpressed, but EIF3 subunits, this E and F are underexpressed. All those subunits are having these initiation factors having multiple subunits ranging from 3 to 8. So you can see EIF3 is all these subunits, if they are overexpressed and this is underexpressed, then cancer can occur also. So this is one example only. What are the? So primarily 10 to 12 initiation factors are required for the initiation process. In normal situation, cap dependent. In case of non-cap dependent, that is during the period of stress, it starts directly at the initiating codon or through the ribosomal, internal ribosomal entry site, that is called IRES. What are that all? Eukaryotic translation initiation is cap dependent, is a little bit complicated, and that requires binding of all those factors. See, normally, this is normally bound, that is forms the normal structure during initiation process, but during ribosomal dissociation, in the main unit there is factor 1, IF1, IF1A and EIF3. These three factors remain attached to it, so that it cannot associate with the 60S ribosome. So with this unit, these are already bound to this 40S ribosome. So to which these factors are also added, and then onto which again other factors are attached that I will discuss, and then there is a poly A binding tail, that cap binding proteins are there, and poly A binding protein, PABP, poly A binding proteins. So they are also required. This binding of PABP is required at the poly A to start initiation. So the initiation, cap dependent initiation, apart from all those factors, poly A binding proteins will also be required. Now, as I told you, this is cap independent, occurs during the process of cellular stress when the overall translation is reduced. Here, it doesn't require cap binding. It enters early at the internal ribosomal entry site, or through a direct binding. So bypassing the need to scan this 5' UTR region. So activation of amino acid is same just like prokaryote. So this is amino acid, tRNA is having one anticodon arm, one is CCR arm. Through the CCR arm, the amino acid is attached to the hydroxyl group. So amino acid tRNA synthesis is there. That enzyme basically is required to convert the amino acid into amino acyl tRNA. That is what is called activated tRNA. And as you can see, ATP is converted to AMP, means two high energy compounds are wasted. One important point, there are two different kinds of tRNA. One tRNA is meant for initiator tRNA, that is called F-met tRNA I, and the other tRNA is for the internal methionine of the protein. Initiation requires recognition of the site. As you can remember, in the prokaryotic site, there is a signed Dalgarno sequence. Here there is also a sequence that is called Kozak consensus sequence. So that sequence is a little around this start codon. So binding this pre-initiation complex I will discuss in a short while. First 40S ribosome makes with the tRNA complex makes a structure of 43S pre-initiation complex. Then when it forms with the binds with the mRNA, then it is called 48 co-initiation complex. And then this initiation starts basically. So you can see here initiating codon in both the cases is AUG, and AUG codes for methionine in mitochondria also. So in case of prokaryote it was phenylmethionine, in case of eukaryote it is methionine. And AUG is identified, you see here the AUG, this is called Kozak consensus sequence. This is the start codon. AUG means +1, this G is +4, this is minus side, this R is basically a purine, in most cases it is adenine, and lowercase letter what you have seen that it is the most common base but variable, means guanosine, cytosine, guanosine, they are common but they can vary. This is basically of uncertain significance also. What is that? So this part you remember what I have written here, that during initiation factor, this initiation factor IF2 and GTP is there. This EIF3, EIF1 and EIF1A is already present in the 43S. So to which basically this EF1, EF4 and EF3 is already attached to these subunits, to which the IF2 M-met tRNA I is known down here. This is initiator codon and 40S. They form, and to which is added EIF5. So that forms basically 43 PIC. That is forming the 43 PIC. Now although I have written the complete structure of this thing, somewhere you might be finding that only 40S IF2 GTP plus 40S ribosome is forming the 43 CPI SPIC, but it is indeed not so, because to keep the 40S ribosome in the dissociated state, all these factors remain attached to it. So these are pre-attached, on the top of which IF5 is also attached, so that they remain dissociated, and that forms the 43 pre-initiation complex. And I am repeating that once this 43 PIC is attached to 4B and 4F and 4B, then you can get 48 SPIC. This is called 48S IC, not PIC. This I have written wrongly. 48S IC. So then the mRNA binds, it forms the 48S initiation complex, and then 60S ribosome binds, complete the 80S ribosome. But EIF1, all the factors on binding with the 60S ribosomes, all the factors which have been attached to it are released, but EIF1A and EIF5A remain bound to the A site, and that needs also released during elongation stage. So this is what I was discussing. See, EIF1, EIF2, EIF3, 43S, and this is the PABP, that is poly-A binding protein, is attached to it, that is basically forming the 40S3S and the initiation complex, and the initiation of protein translation starts. This is AUG codon, and this is 5' end. So this, from this 5' end, there is scanning starts. And what is the Kozak sequence is there? Kozak sequence is just around the start codon. It is almost within the almost start codon itself. Means Kozak colon consensus sequence has reached, means start codon has just arrived. So this is the pre-initiation complex. So factor 1, 1A, IF3, EIF2 met tRNA, BTP tRNA complex, that is forming the pre-initiation complex. The structure was revealed by cryo-electron microscopy. So initiation step, as I told you earlier, during initiation stage, first amino acid will come at the P site. First one. And the subsequent amino acids will come to the A site. Peptide bond formation will occur once second amino acid is attached, that is the start of the elongation phase. So P and R assembly, as I so amino acid is there, the elongation has started. This is another amino acid. So its alpha amino group is here and carboxyl group is here. This amino acid is here and carboxyl group is here. So this basically makes an amino group, makes a nucleophilic attack here, and they form a bond of peptide. This is how a peptide bond is formed, and that is formed while it is within the, this is done by the 28S rRNA, that peptidyl transferase activity is present within the 20S rRNA present in the large subunit. So this peptidyl transferase causes this formation of peptide bond, and this process will continue This process will continue based on the sequence of amino acids which is determined by the sequence of mRNA. Each time ribosome moves to one codon ahead, that is, it is called translocation, and with each movement of translocation, this one unit of amino acid will be transferred to the peptidyl site, the growing chain, and new amino acid will come to this A site and the peptide bond formation will occur. At one stage, this termination codon will be reached. So that will be recognized because there is no tRNA for that. So elongation factors, there are a number of elongation factors, but only two are very important, that is elongation factor 1 and elongation factor 2. So we are having a almost similar type of what I have discussed in the bacterial thermo-unstable, thermostable and elongation factor giant. We are having a homolog. Homolog means this is of common embryonic origin. Analog means it is functionally similar but not of embryonic origin. So, eukaryotic system has evolved from prokaryotes, that is an example. Similarly, eukaryotes having mitochondrial DNA, that mitochondria has been obtained from the prokaryote as a part of evolution, because mitochondrial DNA is having a similar structure of prokaryotic DNA. That I will discuss also. What are the? So elongation I have discussed. The elongation stage will proceed like this. The free tRNA which is present, which will be released every time, will come out from this site only, exit site, and it will make another cycle or another amino acid tRNA formation. So each process will continue with a 3-step cycle. And what are the 3-step cycle? This formation of new amino acid tRNA at A site, peptide bond formation, as I have explained, the CONH group, and then translocation by one codon to the 3' end or to the right side. So P site tRNA is released, the whole ribosome moves to another codon, and this process is repeated until completion. So this is an ongoing process of translation. You can see the sites. So termination. I have told repeatedly there is no tRNA for the top codons. So there will be no tRNA, no amino acid will be placed at the A site. So that release factor recognizes that stop codon. So there are two types of release factor. One is class 1 RFs that bind to the A site mimicking in tRNA. And class 2 is having a GTP activity that enhances the activity of R1, class 1 RF. Now ERF, as I told you, E means eukaryotic. Eukaryotic RF1 is a universal recognized release factor because it can recognize all 3 factors. And ERF will be bound to it, so ERF needs to be released also. So for that ERF3 is required. Mitochondria use bacterial type class 1 RFs. So there are separate genes for these release factors. What are the? So termination will occur no tRNA, so release factor ERF1 enters the site, and this complex releases the peptide chain from the tRNA to the P site. Completed peptide chains are released through a process of hydrolysis. 8Ts ribosome now dissociates into 40S and 60S, and this goes on repeating the another ribosomal cycle. I told you 4 numbers of GTPs are required to make one amino acid elongation. So that is a high energy requirement process. So synthesis means some energy will be required. So once the protein is synthesized, if it is required for this intracellular purposes, maybe in the cytoplasm there is no signal sequence is required. But signal sequence is required if that protein needs to be transported to somewhere else, maybe to the endoplasmic reticulum, maybe to the Golgi apparatus, maybe to the lysosome, then it requires a signal peptide, and the signal peptide leads that particular newly synthesized protein to be transported to that particular subcellular organelle. So type 1 signal peptide is membrane bound. Some proteins are also inserted within the cell nuclear membrane. So these are membrane bound. Type 2 is multi-domain spanning membrane bound protein, and usually it enters into endoplasmic reticulum, Golgi apparatus, or the lysosomes or endosomes, and they follow the secretory pathway to be transported or excreted. So N-terminal amino acid with signal peptide is present in the N-terminal end, and that needs to be cleaved to make the functional protein. That is one example is insulin. I am telling you, this insulin is synthesized at a P-pro form insulin because it is also having a signal peptide, and that allows it to enter into the endoplasmic cistern, and in the endoplasmic within the endoplasmic reticulum cisternae, it folds onto itself to form an A chain and B chain, and they are cleaved. One important point again I am telling you, disulfide bond formation of protein cannot usually takes place within the cytosol because of high amount of glutathione. Glutathione reduces disulfide bonds, so intracellular bonds can form only when it is not within the cytoplasm, within the cytosol basically, because glutathione is not present. See here, this pre-pro-insulin is entering into the endoplasmic reticulum, where it is folding onto itself to make disulfide bonds and intramolecular bonding also, and with the removal of the signal peptide and the connecting peptide of the insulin, the 2-chain insulin, that is A chain and B chain, A chain containing 21 units, B chain containing 30 units, they are having one intra-chain disulfide linkage and 2 inter-chain disulfide linkage. That active form of insulin is released. So you see here, this is synthesized in the ribosome. The signal peptide is here. Through the signal peptide is entering into the ribosome, into the cisternae of the endoplasmic reticulum, where it is folding onto itself, and then it is transported to other organelles. So single chain precursor molecule following an intra-chain disulfide bond. What is that? So post-translational modification. So in prokaryotes, it is basically polycystonics, means multiple proteins are formed from a precursor molecule, and they are cleaved at a particular point to release the particular protein. So there is no post-translational modification is required in case of prokaryotes. But in case of eukaryotes, it is usually monocystonic. Monocystic means only one protein is synthesized at a one point of time. So that protein may have a leader sequence, may require post-translational modification depending on what type of protein it has produced. So it undergoes several post-translational modifications. Since the time is almost 11 o'clock, I stop here and I will discuss post-translational modification in the next class, and also a little bit of mitochondrial translation. So meanwhile, I want that you read this topic, both the topics, so that you understand better. I can have a comparison between the prokaryote and eukaryote as well as mitochondrial translation, and then I will move on to the next topic. Okay. So go and read. That's all I can say. I have seen most of you are engaged as usual in the gossiping. You are not serious at all. One point of time, within a month, you will be required to come to the stage to deliver a lecture like me. At that time, you will get an interest of what I am feeling as a teacher to you. You are gossiping, and we all faculty sit there and start gossiping. You will feel the same thing. And at least I am seeing very clearly that nearly 80 to 90% students are not listening to at all, and they are engaged in some other more important activities. Please continue with your own activities, because you are already an adult, and I have told you repeatedly about this bad habit, and you are not in a mood to stop it. So you will face the consequences one day. That's all I can say. Chalo.